Let us repeat this same figure and let us do some other consideration. We have seen that SOX10 regulates the expression of the Schwann cell-specific myelin genes PZ, MBP, and PLP. I don't stay here to express them in full because now we are familiar with these acronyms. And it also regulates the expression of connexins in the peripheral nervous system myelin. And we have other types of connexin that we met before in kind of general explanation of these proteins. And these are the connexin 32 and 47. In the autonomic nervous system, SOX10 regulates, as we have seen, the expression of MESH1 and FOX2B transcription factors necessary for sympathetic nerve fate. And again here we see these unknown acronyms that we are going to explain more diffusely. In the final note here, in the enteric nervous system, SOX10 interacts with PAX3 to activate the GDNF-CRET pathways, and regulates also the endothelin receptor beta during the enteric nervous system development. We have already met all these things, we have already explained all these things. Now let us go to see what they mean, or the meaning of these acronyms. The details of the upper text are following this line. So myelin protein 0 or PZ or MPZ, P0 or MPZ is a single membrane glycoprotein which in humans is encoded by the obviously homonym acronym MPZ gene. Mutation in myelin protein 0 is important because it can cause myelin deficiency, and it is associated with different kinds of neuropathies, like the Charcot-Marie-Tooth disease and the Dejerine-Sota disease. The Charcot-Marie-Tooth disease is a hereditary motor and sensory neuropathy of the peripheral nervous system, characterized by progressive loss of muscle tissue and touch sensation across various parts of the body. The disease is unfortunately quite common, involving 1 in 2,500 people. Currently, unfortunately, there are no cures, no treatments for these disorders, with care maximally focusing on maintaining functions. This syndrome was previously classified as a subtype of muscle dystrophy. The Dejerine-Sota disease has many names, such as the Dejerine-Sota neuropathy or progressive hypertrophic interstitial polyneuropathy or others. And there is a hereditary motor and sensory polyneuropathy of type 3 or Charcot-Marie-Tooth disease of type 3. And these again here hereditary neurological disorders characterized by damages in the peripheral nerves and resulting in progressive muscle wasting. This condition is caused by mutations in various genes and in P0 and MPZ or MPZ protein. Then let's go on and let us see the importance of the MBP protein for myelination in the nervous system, for both myelinating oligodendrocytes and in the central nervous system and Schwann cells in the peripheral nervous system. And the proteolipid protein, that is a major myelin protein from the central nervous system. It is crucial also in the formation and maintenance of the multilamellar structure of myelin in the Schwann cells and the oligodendrocytes. Then there are the connexins that we met before, or I remember gap junction proteins, and I don't go on in explaining them because we have already seen their fundamental role in vertebrate gap junction in developing astrocytes and in other cells. Let's go on with SOX10, that is a very complex factor. In the autonomic nervous system, as we saw before, SOX10 regulates the expression of MESH1 and FOX2B transcription factors necessary for sympathetic nerve fate. In the enteric nervous system, the ENS, as before seen, SOX10 interacts with PAX3 to activate the GDNF or CRED pathways, and regulates also the endothelin receptor beta during the enteric nervous system development. MESH1 now is reclassified as ARC8 skewed homolog 1, is a protein encoded in humans but the ASCL1 gene. This protein plays a fundamental role in the neuronal commitment, that means in the destiny definition of the different neurons and the typing of these neurons and their differentiation, in the generation of these neurons also as olfactory neurons or autonomic neurons. FOX2B is a protein that in humans is expressively exclusively in neurons of the visceral compartment, the cardiovascular, digestive and respiratory system, and is required definitely and most importantly for their differentiation. PAX3 has the name coming from paired box, PA initial and the final X PAX genes, that is a family of genes coding for tissue-specific transcription factors. All the transcription factors that we saw before, containing an N-terminal paired domain, and usually a partial, or in the case of 4 family members, PAX3 to 7, PAX3, 4, 6, and 7, that are a complete homeodomain to the C-terminus. An homeodomain or homeobox is a DNA sequence of around 180 base pairs long, found within genes that are involved in the regulation of parts, of patterns, of very big sections of anatomical development in the morphogenesis diffused in the whole living world, and specifically in the human embryos. Let's skip now to the other strange acronym PDGF and PDGF, that means platelet-derived growth factor. Why platelet? Because it was observed in the first stages of its observation in platelets, but they are diffused in being crucial factors as power mitogens for cells of mesenchymal origins such fibroblasts, smooth muscle cells, and it is to be put here a very special attention to glial cells. So glial cells share their destiny with a number of other different cells, such as fibroblasts or smooth muscle cells. And this means that all the cells have common factors regulating their growth and are deeply interlaced in their destiny and in their future development and functions in the full developed bodies. Here we can see in the red rectangle the whole machinery of a PDGF factor with a receptor in the EGFR receptor, GPCR receptor, and other channels. And as we can see here, there are extremely complex transmembrane proteins interacting among them. And then we have the CSPG4 or NG2, also called chondroitin or chondroitin sulfate proteoglycan, also known as melanoma-associated chondroitin sulfate proteoglycan, NCSP, or, pay attention, neuron glial antigen 2, NG2, that is a chondroitin sulfate proteoglycan, a complex glycan molecules, that in humans is encoded by the CSPG4 gene on chromosome 15, as we can see enclosed in the red rectangle on the right figure here, showing the human chromosome in order from 1 to the final Y chromosome. CSPG4-NG2 is also a hallmark protein of oligodendrocyte progenitors, and it is mostly important because we know now that this factor, this NG2 factor, is crucial for oligodendrocyte development from their precursors, from the OPCs. OPC's dysfunction has been implicated also as a candidate. It is not the only candidate, but it is supposed to be crucial in pathophysiological mechanisms of familial schizophrenia. Familial schizophrenia has different types of clinical manifestation depending on the person's symptoms, and there are main types of schizophrenia, including paranoid schizophrenia, catatonic schizophrenia, disorganized or hebephrenic schizophrenia, and residual or undifferentiated schizophrenia. I don't go in depth in these descriptions, but it is enough to have well in mind that schizophrenia is a constellation of nervous system disorder. And here we show the BKS1, also brain-enriched myelin-associated protein 1, that is a protein coding gene with disease associated with its anomalous differentiation or anomalous section in breast cancer and in cerebral creatine deficiency syndrome 3, that are very rare but devastating pathologies. BCAS1 or brain-enriched myelin-associated protein 1 expression identified newly generated myelinating oligodendrocytes in multiple sclerosis lesions. And see in the A figures where there are the violet designs, the confocal images of this gene, and in this case of the protein, where the oligodendrocyte transcription factor 2 is signed instead in the second figure in white, and they are present in the corpus callosum at the postnatal day 40. So we have BCS1 and oligodendrocyte transcription factor 2 or OLIG2 that are cooperating in these two stages of the brain development, and namely in the cerebral creatine myelin-associated protein and in the oligodendrocyte development, especially in the corpus callosum, in the late stages of the postnatal oligodendrocyte maturation. Let's go now to the ENPP6 or ectonucleotide pyrophosphatase phosphodiesterase family that is highly expressed in liver sinusoidal endothelial cells, and as a rule we see them also in developing oligodendrocytes, which actively incorporate choline and synthesize PC, phosphatidylcholine, that are crucial substances involved in oligodendrocyte development. And put your attention to the figures in D and in E. In D we have an immunohistochemical analysis of the ENPP6 developing in the mouse brain from P2, P4 to P12 and P14 postnatal days. And we can see indicated by the arrows the position of these factors. And note that in the first stages, these factors are localized around the nucleus at P2 and P4 stages. They are not within the cells, but in the external part of this oligodendrocyte, by these liver cells, but it is the same in the oligodendrocytes. And as we can see, these promoting factors are getting within the cells, and we see very easily that in the figures in P12 and P14, ENPP6 is strongly expressed in the myelin sheath, but rarely in the cell bodies or perinuclear region at P12 and P14. In E, we have the protein during the differentiation of the precursors of oligodendrocyte precursor to oligodendrocytes fully blown and fully developed at different hours during their development. In this case, it has been made a special treatment, but it is not important. We must look at the black spots, and as we can see, they become bigger and bigger during the passing hours during the development, the postnatal development of the oligodendrocytes. And as you can see, at the 0 hour, there are no expression of this substance, of this protein, but at the 96th hour, that means the 4th day of development, they are extremely strongly expressed in the cells of the oligodendrocyte, and they participate actively in the oligodendrocyte definite and definitive modification. And let us see just here, just to have a memory of molecules we all have studied in our biochemical courses, the galactosyl ceramides and the sulfatides. that are expressed here in a very simple metabolic pathway during the sulfatide metabolism. And we can see that from serine and palmitoyl coenzyme A in different stages of development, we can arrive to the final sulfatide or 3-O-sulfogalactosylceramide stage, or fully developed sulfatide in the final stages of the glial cell membrane. And we can see the role of these sulfatides in healthy conditions and in the myelinating disease. We can in some way mimic the myelinating disease indicated by the red arrows that goes from ganglioside to sulfatides, and this show the complex metabolic cascade that are called in to reintegrate the myelin axis and sheathing. In fact, they go as they can from ganglioside, trying to reproduce again the necessary sulfatides to regain the integrity of myelin, while vitamin K acts as an opposite factor, bringing the sulfatide molecules again to gangliosides. And the red path is the path that the sulfatides try to afford and to go through to get again a correct myelinization during the remyelination stages in the myelinating disease. As quoted in previous slides, two parameters are critical in the oligodendrocyte development: the modulating factor of temporal organizations and the precursor, oligodendrocyte precursor, permissive or blocking roles of the factor involved. And then let us see the different developmental stages from neuronal progenitor cells to myelinating oligodendrocytes. The modulating markers and their temporal organization are shown here. As we can see, we have the neural precursor cells in violet, and then we have the oligodendrocyte precursor in the next stage, the pre-oligodendroglial cell, and the final well-developed oligodendrocytes in the final, the right part of the figure. And we have stripes in the bottom part of the figures with the acronyms that we now know very well, and they indicate the intervention times of these factors during the development from the neuron precursors to the full developed oligodendrocytes. And as we can see, all these factors intervene either continuously, as SOX10 for instance, or in special stages of this course, of this trail of development of the oligodendrocytes from the NPCs. And as we can see, OLIG1 and 2 oligodendrocyte cell lineage markers and SOX10 and NKX2.2 are expressed in all cells of the lineage, the three green stripes, while the A2B5 factor recognizes the progenitor cells, the NPCs, and helps them to differentiate in oligodendrocyte progenitor cells, OPCs, as we can see in the yellow stripes underlining the transition from MPC to OPC. then these factors disappear and appear other factors that are the PDGFR alpha and NG or NG2 markers that characterize the OPC development to pre-oligodendrocytes stages. And in fact, the two light maroon or ochre stripes underlie the development and the path from OPCs to pre-oligodendrocytes, the two red cells in the midst of the cell line showing on the top of the figure. And then we have a scaled intervention of other factors. And it is very interesting that they have right times in the right cells, and this is illustrating the extreme complexity of the development of oligodendrocytes. And as we can see, PLP 04 or 04 or 1 and CNPAs distinguish transition from progenitor of oligodendrocyte, pre-oligodendrocytes to full developed oligodendrocytes. And they are scaled in these interventions to get to the final stages. The MBP, the MAG, MOG, and GALC modulating myelin protein expression of differentiating axon-myelinating oligodendrocytes. So we have a true orchestra of cell lineage markers and the progenitor maturing factors from MPC to a full developed oligodendrocytes. And then let us see now the complex problem of the ontogenetic opposite effects. That means pro or opposite development factors. And these factors have their effects in embryonic time or in the postnatal stages. That is well resumed in this scheme that I am going to show you, that put in mind it is extremely simplified and very incomplete. As we can see here, we have extracellular elements playing a permissive promotional pro-neurogenic factors, pro-neurogenic actions on the top part of the figure, and they are signed in green. And they are an extremely great number of factors getting from proliferation recruitment stages to full myelination. And we have inhibitory, antineurogenic factors in amaranth in the bottom part of the figures, contrasting the permissive promotional neurogenic factors we showed before to get the oligodendrocyte development and their final stage of mature myelinating oligodendrocytes. It is extremely important to note a further complicating issue, that in some cases the same molecule may exert inhibitory effects at one developmental stage and promotes a promotional of permissive role at other stages. And it is extremely important that they intervene at the right moment of the ontogenetic development, because wrong times of intervention bring to severe diseases during the development of oligodendrocytes. Let's have some interesting note on the metabolic and energetic elements of the oligodendrocyte development. During myelin biogenesis, oligodendrocytes synthesize, as we have already seen, considerable amounts of myelin components like MBPs, PLPs, galactosylceramide, and cholesterol. These external substances can take up to 100 times the weight of a single oligodendrocyte, and it is extremely important because they give the oligodendrocytes their final shape and their final volume. In fact, oligodendrocytes have the highest, put your attention to this fact, the highest oxidative metabolism of all the cells in the central nervous system during active myelination. So oxidative metabolism that is considered, has been considered a very negative fact for producing oxidative substances until recent year. In very recent works have been reconsidered as powerful positive factors during definite metabolism stages. And the other phase of this medal, with the other highest oxidative metabolism that contributes to the accumulation of the MBPs, PLPs, galactoceramide, and cholesterol of the first stage, the oligodendrocyte precursors have very low levels of antioxidant, such as glutathione, very low levels of these antioxidants during this development. And it is a very delicate fact because the features of very low antioxidant factors like glutathionase might explain why myelinating oligodendrocytes are exceptionally vulnerable to metabolic stresses, possible contributing to the multitude of pathologies involving demyelinization. This is a crucial factor because we have the need to have the highest oxidative metabolism to allow to oligodendrocyte precursor to be covered by the MBPs, PLPs, and other substances to become the big cells we have seen. And in the meantime, they transit during a very delicate phase of their development, getting the path to the possibility of metabolic stresses and to further pathological stages of demyelination. Now let's consider the gross embryologic and anatomofunctional topographic migration and spatial configuration of oligodendrocytes in the brain. We have seen many features of glial cells, factors facilitating or opposing their development from their precursors, and other stages of maturation of this kind of cells. Now let us see where they are exactly in the developing brain. Let's now go to the oligodendrocyte maturation process from the precursors of oligodendrocytes in their progressive myelinating stages of the cerebral nerve fibers following three waves or three stages. The precursors of the oligodendrocytes are derived from neural progenitors of radial glia. In this figure, we see in A the full three-dimensional representation of an embryo, a mouse embryo in this case, with the telencephalic vesicles with two bluish rectangles indicating the cutting section of these of this structure. Just on the immediate right, there is a longitudinal section of the brain, of the developing brain, with different areas that are imaged with LGE, MGE, AAP, and CGE. And the third stage, the third figure here illustrates in a laying position the complex structures of what we illustrated before by the acronyms, showing the green, the reddish rose section and the violet section that are fundamental sources for the migration of oligodendrocytes precursors. In C, we have the mature adult mouse brain with again a rectangle indicating the section level, the coronal section level of the brain. In the lower part, we have the appearance of the different sections at different embryonic ages, at E10, E13, E16, and in the adult configuration. And we have seen they are populated by differently colored oligodendrocyte maturation. And in the adult brain, we can see a mixed and very complex array of oligodendrocyte maturations. The precursors of oligodendrocytes begin to develop at day 11.5, and for some day this first wave goes on getting to the goal of maturation for the second wave that we will see after. This first wave of precursors emerges from the medial ganglionic eminence that is written here in red, in the red circle onto the sagittal section, and that is a transitory structure in the development of the nervous system that guides cells and axon migration. And again from another section that is the caudal ganglionic eminence, but overall, most importantly, there is the anterior entopeduncular area that is assigned by the purple circle that is assigned here in AEP. These are particularly important initial stages that give rise to the second wave. The second wave, represented here, starts at the age of E15, the embryonic age at the 15th day, from the lateral and or the caudal ganglionic eminences that are represented here by the LG circled by the green circle in the image, and by the CGP or caudal ganglionic substance into the maroon circle, or the amaranth circle, as you like. And OPCs that emerge from both waves, the first and the second, populate the murine cerebrum in a ventral to dorsal manner. And we have here the third wave, where OPCs are generated in the first week after birth, postnatal, originating from the dorsal cortex. And we can see here the population of these astrocytes and oligodendrocytes, the radial glia, that goes to densely occupy the superficial cortex, as it is indicated by the maroon round rectangle represented here. PCs that are derived from the first wave disappear after birth and are virtually undetectable in adulthood. We now face the subsequent myelination processes, a complex with a synchro-diachronic and geometric spatial dynamics. It is a very complex process, and we will see it in an analytic way. As first, we must consider the OPCs proliferating and migrating towards naked axons. And then we know that OPCs differentiate into pre-myelinating oligodendrocytes, and they extend multiple processes that go in the environment, contacting axons, but not yet starting a myelination process. At the nearing of oligodendrocyte prolongations or processes, with many primary, secondary, and tertiary processes, when they get near to axons during the myelination process, this kind of dynamic does not start immediately, but only after silencing of mainly axon-derived inhibitory factors for oligodendrocyte differentiation. Then the pre-myelinating oligodendrocytes, after getting in contact with these inhibitory factors, retract their secondary and tertiary processes, and myelin membranes become ready to be elaborated by the primary prolongements or primary processes when getting in touch with axons. But this process is really more complex than we can think, and we will go to analyze it in the next slides. The regulation stages of CNS myelin targeting the surrounding condition of their correct emergence. This is meaning that there are many conditions that give a very variable result in the myelinating process. Not always when a primary process from an oligodendrocyte getting in the nearbys of an axon terminates in a correct actual myelinization process. There is a list of different factors influencing these kind of actions. First of all, the importance of mutual geometric relationships between myelinating probes, the primary oligodendrocytic process, and their targets. That this means the new axons to be myelinated. So we can see a quite complex image here, where in A, B, C, and D are reported many conditions, factors, events that preside to the full myelinization of an axon. And as we can see in A, we have an oligodendrocyte precursor, the OPC cell shown at the top left of the figure, that in days and up to months, go through a selection stage, and this selection stage has a duration of hours or days, and goes through the prolongation of many processes, first primary, secondary, tertiary, and so on processes from these precursors, and then a primary establishment of this new oligodendrocyte, or the oligodendrocyte primary oligodendrocyte, or new OL, getting in touch, trying to establish connection with the axons in the nearbys. And in few hours, this new oligodendrocytes begins to establish a myelin body, the myelin body, or ensheathing tract around the axons. And as we can see, we have axons, thick axons, the gray line in the bottom of this small image, or more subtle, with more subtle neurites. And this is the beginning of the refining stages. And in the refining stages that have a very different duration, we can observe the elongation of this covering by myelin on the axons, the loss of myelin degenerative processes that are not chosen by for the final myelination, and the final myelination stages with the green thick lines indicating the myelination process. And so there are the two process stages, the selection and the refinement stage. And as we can see in B, there are factors indicating different biophysical properties of the myelinating or being to be myelinated axons to grant for a correct myelination. First of all, there is the increasing or decreasing curvature of the axon. The axon in B are represented by the blue membrane within the inside of the axon, indicated in bluish, light blue color, or there is the body of the neuron represented as a sphere. And we have elements with very different diameters and very different curvature. Obviously, the thicker is the axon, the minor is the curvature. And we have an increasing left-directed arrow trying to show, to describe the increasing curvature of neurons that can arrive down to 0.1 micron in diameter, or to bigger axons reaching even one or more microns in diameter. And then we have the bodies of the neurons that can get the diameter of 10 to 20 and even 30 microns, not shown here the 30 micron image. And this increasing diameter of axons or of neurons is a crucial factor for permissive or repulsing the myelination process. We can see some example in C, where we can see in the first part of the figure there are curvature sensing mechanisms. And we have in the first section of the figure a kind of mismatch between the terminal in green of the myelinating oligodendrocyte and the axon surface. In this case, there are many processes leading to the retraction of this prolongation of the oligodendrocyte, and the oligodendrocytes detaches from this primary contact and goes to find for other targets. In the second section of this figure, we can see a perfect match between the surface of the oligodendrocyte process and the surface of the neuronal membrane. And in this case, as indicated by the green arrow, we have a kind of mutual recognition of the optimal conditions for myelinating the axon. A second factor is given by the stabilization process that is regulated by length sensing mechanisms. What does it mean? We have a coronal section of an axon, the black circle with the internal bluish cytoplasm of the axon, and the green with orange dots stripe getting around the axon itself. And as we can see, we have a too long process of oligodendrocytes that does not allow for a correct myelinization process. While in the second figure, we have a correct length, mutual accomplishment of length of the oligodendrocytes and of the axon diameter. And this allows for a correct start of the myelination process. There are also, and this is called the curvature or length sensing proteins on the surface of the two elements, the axon and the oligodendrocyte process. And in D, we have a list of proteins or glycoproteins that act as cell adhesion molecules on potential targets regulating the CNS myelination. And we have positive regulators and negative regulators. And as we can see, we do not go through the single elements. There are many factors that either permit and stabilize the process, or and they are positive regulators, or they have a negative action on the attachment of the elongated process from the oligodendrocytes on the neurite membrane. And finally, we have again a very important event regulating the attachment of the oligodendrocyte process to be either attracted by the neurite surface or repelled from this surface. And we have here represented the very tip of the oligodendrocyte process that expresses a special protein that is called ephrin, that must match with the surface positive regulators on the neurite membrane. And in this case, the attractive targets in green match perfectly with the ephrine expression by the oligodendrocyte process, and the myelin sheath can begin to attach to the neurite itself, while the opposite happens in the case of repulsive targets. Now let us see the different roles of oligodendrocytes in pediatric and adult humans with the list of pathological signs that are evident in experimental animals. These are called the whole series of events, a kind of the description of the roles that is containing in the definition of gene ontology in the OPCs, in the pediatric human brains, that is related to oligodendrocyte differentiation, the extracellular matrix metabolism, the exon guidance, and the cholesterol transport. We will see better what a gene ontology is in the next slides. And we have also a gene ontology in precursors of oligodendrocytes in adult human brain that is related to regulation of cell projections, regulation of molecular transport, and the production of superoxide metabolic elements. In addition, we can see here that as observed for now only rodent adult OPCs, but it is arguable that they are active also in the mature brain. In the aged CNS, we have increased the DNA damage and decreased metabolic functions in aged rat or mice, and fail to respond to differentiation signals, both in vitro and in vivo. This may underline the poor remyelinization observed in aged rodents. The signs may suggest potential metabolic pathways that in demyelinating disease could occur in the oligodendrocyte homeostatic regimes. As I told before, we go to explain what an ontology is. An ontology is a formal representation of a body of knowledge within a given domain, of a knowledge domain. Ontologies usually consist of a set of classes or terms or concepts that are related among them, with relations that are operating and active among them. The definitions comes from a much wider field that is the philosophical field. In philosophical terms, an ontological approach conveys search for an answer to the existence of an object as in its relational identifiability of the object itself. When we see an object, whichever object we are talking about, that is abstract or true, we can establish and identify its relational field, or to which other objects the single object is related to. And this is called in some way the ontological field of the object. The gene ontology, instead, describes our knowledge of the biological domain with respect to three aspects: the molecular function, the cellular component, and the biological process. Now we see the myelin features. I show here a very complex image, and people who is bored or considers these explanations a bit too heavy can skip this section, this figure, and pass to the next slide. Anyhow, going on with the explanation, we have a very natural imaging of the myelination processes that happens also in cultures, in cell cultures, just because we cannot observe them in the whole brain in the animals or in the embryos of animals. In A, we have an in vitro co-culture system indicating the myelin formation. And what does it mean, co-culture? This means that we make, very simply, cultured cells. We cultivate the cells in vitro, and co-cultures means that we cultivate two types of cells: the neuroblasts, becoming very fastly neurons, and the oligodendrocyte precursors. And you observe the dynamics that the oligodendrocytes and the maturing neurons are going toward, in order to be able to be aware of the extremely complex stages this myelination process presents also in vitro. And just in A, we have blue brackets that mark regions that present just in the figure with MBP, myelin basic protein, show the specificity of this myelin basic protein on the axons. And these are the proto-stages, the primal stages of myelinization by an oligodendrocytes. The white spots or slightly elongated spots under the blue brackets indicate the blocks of myelin that go around the neurites or the axons during the development. And the white indicators is special indicators from the membrane basic proteins, and it appears as a kind of white or white-gray spotting of this protein along the axons. And in the nearby figure, always in A, we can understand that there are many other colors, and each of these colors indicates a special protein. And we can see here in the second figure that the blue brackets identify the two main proteins involved in this, that identify in gray the membrane basic protein of myelin, and with the red, the green colors indicates the neurite specific protein called class 3 beta tubulin that we have already met before. In this case, TUJ1, class 3 beta tubulin of type 1. So we have marking regions indicating myelin, that is tubes of oligodendrocyte extension wrapping the tubulin positive neurites in this co-culture. And if we observe well the figure B, we see on this extreme left the dotted lines getting and converging in the two groups of dotted lines converge towards a specific region within the A figures, the colored A figure. And they identify the neurite with oligodendrocyte wrapping them regions that have been transected in the next B figure, and we can see that it is a kind of slice of this prolongement, identifying in white the membrane basic protein, and in green it identified the tubulin expression with the green acronym. And we can see that getting together the two dyes, they create a red image. And the red image says that the two classes, the two types of proteins are strictly connected among them. Then in B, let's go to C, and we have a transmission electron microscope, a classical flat electron microscope, to be distinguished from the scanning electron microscope that gives very important and very interesting three-dimensional images, and we are not showing them here. And as we can see in C, these 10 images show the compaction, that means the step-by-step aggregation of the oligodendrocyte membrane as a glial membrane around axons to form the myelin wrap around the axon. And in D, we have the two examples of the myelin basic protein and contacting associated protein localization during myelination of mixed co-cultures. There is not too much to say again on these figures that should be described much more in detail, but I wish to show you that all the proteins, the substances and the structures like oligodendrocyte precursor and neurite structures are really present always, even in vitro cultures. Let's skip to the next figure that is much more simple by luck, and it is a model for myelinization, because we must get to the final stage of a kind of summary of all these kind of events, elements, substances and structures that are involved in myelinization. And as you can see from the figure, within the squares of this matrix, there is a upper medium square with the word survey, then a lateral square in the second row with the name of anchor, then a third image with W wrap, and the final last row in the mixed figure with the word expand. And these are the 4 stages of the Sarapi model, and we are going just now to understand what does it mean. SARAPI is an acronym that means survey, anchor, wrap, and expand. That, if we put attention, are the four words that we enlightened before within the figure. And the SARAPI model describes the oligodendrocyte progressive myelination of axons as in 4 stages, with the checking stage where the process of the oligodendrocyte gets near the axon and checks it. We have seen all the methods for the checking, with repulsing or accepting molecules, with diameters, shaping, and so on. Then there is a second phase, that is the anchoring on the axon. If, for instance, the terminal of the oligodendrocyte process has met proteins like ephrins or the right diameter of the axon, it anchors. And there is an anchor point, as we can see from the figure, where the axon attaches to the axon. And then we have a third stage called the W-wrap, that means wrapping, that is a turning around of the process, of the oligodendrocyte process, turning around the axon, the neurite. And we have many turns and turns and turns of wraps around the axon. Finally, the oligodendrocyte membrane, in the expressing this myelin mancot, expands in width and becomes a very long myelin sheath covering the axon. And we have the section in the very last figure on the top right of the matrix of squares, showing the section of this neurite enwrapped by myelin, where in red is represented the neuron and in green is represented the myelin in the transverse section, hypothetical transverse section of this myelinated axon. And then let's get to the final stages of this section on oligodendrocytes, and let us some consideration on extrinsic mechanisms of myelination support. Oligodendrocytes support axonal transport and maintenance via exosome secretion. Neuronal axons need long-term integrity that depends on intrinsic mechanisms such as axonal transport and extrinsic support from adjacent glial cells. So we have a double set of factors and substances that maintain well-functioning the oligodendrocytes. Some factors are purely intrinsic, generated within the oligodendrocytes. Some others are external. And one oligodendrocyte extrinsic, external mechanism to support neurons is represented by the production of specific extracellular vesicles called exosomes. What are exosomes? They are lipid bilayer delimited particles naturally released by a cell. And we can observe the generation of the structure of an exosome. Exosomes are vesicles within the cell, and we can see in 2 parallel curves curved lines indicating the membrane of the cells, and these phospholipid bilayer membranes contain DNA, proteins, mRNA, and pure RNA, and they are not able to replicate themselves, but they are fundamental to support the well-being of the oligodendrocytes receiving them, because they enclose their exosomes, and the exosomes are very easily engulfed into the cell membrane because are formed like cell membranes, and they open very easily on the cell membrane, getting near the membrane itself and releasing those factors within the receiving neurite covered by the glial cells. The exosomes, as we have seen, contain a large array of substances that act as and of molecular cargoes that are useful to the metabolic state and promoting the axonal transport. Mutations in oligodendroglial exosome secretion show secondary axonal degeneration due to oligodendrocyte-specific gene defects. This is extremely recent research appeared in this December 2020, and it represents a very crucial step to understand not only the internal factors or the protein as we have seen, but complex bodies as exosomes supporting the oligodendrocytes' well-being and correct function. Let's go to the final stages of the myelin-neuron interactions. This is a quite complex figure, but if we analyze it in details, in steps, all things will appear quite clear. We can see here on the top starred figures that represents the neuron body, and then we have the axon with the stripe in violet with many different structures constellating its surface, and the orange and the red little spots represents the ionic channels, and these are represented almost only sodium and potassium channels. Sodium and potassium, I remember, to those of you who have potentially forgotten their roles, are the starting enzymes for the production of spiking actions from the neurons, the electrochemical activity of neurons. The green structures with these strange forms are the myelin sheets enveloping with different length or different thickness the tract of axons interested to myelinization. And we have different degrees of myelinization. We must remember that neurons are at least, in a very simplified way, in three classes. There are nude axons, axons without any myelin coverings, and there are different degrees of myelin enveloping, called with different acronyms as delta neurons, beta neurons, and alpha neurons. And we will see the extreme importance of these definitions. The nude neurons are called also C neurons. Curiously, they transport, for instance, noxious stimuli from the periphery to the central nervous system. And going on and forward in the different degrees of myelinization, we have the A delta lightly myelinated fibers, then the beta fibers with a higher degree of myelinization, and the final alpha neurons that are very, very heavily myelinated axons. And we have also the terminal of this long axon with the synaptic structures indicated by black dots in the top, on the top of the figures, getting in touch with a second neuron that is again represented by a star, but this means that the main regions of the neuron where the synaptic terminals of one neuron terminate is represented by the dendritic population. So let us give a glance here to the axonal subdomain and the amount of glia and the importance of the amount of glia around the axons. We have in number 1 in the upper figure and in the lower part we have one stripe in this kind of matrix that is indicated as axon initial segment, ACE. This axon initial segment is also called the axon hillock. It is generally not a straight line like here, but it is represented by a converging piece of the double membrane in a kind of arrow getting within the stripe of the axon. So this axon initial segment or axon hillock has many different properties that because it can change its position, as we can see in the difference of figures between the 1A and YB figures, reporting the different sodium and potassium channels. The little v in the nearby means voltage, but we are not looking at this particular now, and can alter its length, so, and its ion channel density. In fact, in 1A we have a high density of ion channels. In the B part of the figures we have a scattered density of ion channels. And in C another configuration with two different sections of these axons. Sorry. These three configurations modulate heavily the cell excitability, the AP shape, that means the action potential shape, and the frequency firing of the neurons. Then we have to consider the myelin sheath that is represented by the number 2 in the upper figure and the second row in the matrix below. The myelin sheath can be characterized by different lengths and thickness and by the number of wraps around the axons. What does this mean? That the myelin sheath these wrapping myelin sheets identify tracts of neurons that are completely covered by myelin, and in these tracts of neurons, of axons, for better definitions, are not presenting an electrochemical exchange of ions to governate, to regulate the cell excitability and the transition of the electrochemical signal. But on the opposite, in the long thread of the 2A figure, we have a channel-free segment, and the transition of signal is purely electrotonic. What does it mean? That are electrons starting from the one side and getting to the other side of the myelin sheath protection zone. And as you can understand, the ion, sodium and potassium governed electrochemical events takes time to become active, and the transition of the electrochemical signal or the spike of the neuron is quite slower than faced to the myelinated tract of the neuron. And there is an interesting phenomenon. We could have a small degree of myelinization, as in 2A, and this small degree of myelinization often coincides with a minor number of wraps and also a minor length of these wraps around the axon. So we will have short segments of myelinated segments of the neuron. And in 2B, we see a much higher degree of myelinating zone that is lengthier than the previous segment, even if it doesn't appear here, and is much thicker. And the lengthier is the zone covered by myelin, and the faster will be the conduction of the neuron, because the tracts of axon covered by myelin are longer, and the electronic conductive transition of signals is much faster than the ionic generated electrochemical signals. So the myelin degree shapes the conduction velocity and the frequency firing, as well as the context of the action potential shape and the metabolic support. How these signals running within the tunnel generated by the myelin sheets around the axon can proceed if these wraps of myelin are not completely covering the axon, but are segmenting it, because they are not continuously wrapping the axon, but they terminate every such time to get a free zone of the axon. In myelinated axon, there are in fact short tracts of a nude axon that are called the nodes of Ranvier. And these nodes of Ranvier show an electrochemical conduction of signals. Why this happens? Because in the tracts covered by myelin, the electrotonic conduction is definitely very fast because it's conducted by electrons, but it decays in time, and it is a very well-known physical principles, as it is known, for instance, in the submarine ropes conducting radio signals. And it was a discovery of Lord Rayleigh in the second part of the 19th century. And the fact that the electrotonic signals decay along the tract of the myelinated axons, this means that they must be revived in some way by this zone of electrochemical exchange or electrochemical signal generation. These electrochemical tracts are a kind of batteries reviving the decaying electrotonic signal. And as we can see in ABC, we have different kinds of nodes of Ranvier because they can be lengthier or shorter with the changing ionic channel concentration, as it is evident in A and B. And the same node of Ranvier can change, as in 3C, its shape, becoming a bit enlarged or more restricted in its shape. All these features are influencing the conduction velocity and the action potential, the AP shape. Then there are several other features that, like the so-called paranodal regions. The nodal paranodal regions is the confined region between myelinated and unmyelinated parts of the axons. And there are, as you can see in 4B, black structures connected by wired proteins that are called the paranodal proteins, and they are also modulating the action potential shape and the cell excitability. So myelinated sheets are governing the conduction of neurons, not only by the electrotonic conduction, but also on these very delicate regions of paranodal proteins. Within the nodal paranodal region, the length and the ion channel density of the node of Ranvier may change upon changes of myelin in A or via inside-out mechanism, as in 4B, whereby the axonal cytoskeleton fine-tunes the morphology and composition of the region, as we have seen in the heavy green little rectangles connected by these strange helicoidal proteins. Then there is another factor, the axon itself, that can vary in diameter, as we can see in A and B. And in B, we see that the axon can show either tapering shapes or enlarging shapes, changing completely the electrochemical conduction velocity of these kinds of tracts of axons. And they have also a differential ion channel density, and obviously they interfere and modulate, as all the other factors, conduction velocity, AP shape, and frequency firing. Then we have a terminal part where there are the synapses toward the other neurons that can be populated in the very terminal parts. They are nude, they don't show myelin sheets, and they can present richer or poorer populations of ions, changing at this heminode, as it is called, because we have not another section covered by glia but we have the terminals with the presynaptic terminal of the neurons. They shape, even in this case, the conduction velocity. And we get now to the final consideration, where we put together oligodendrocytes and astrocytes. And this is the complex background of the interrelationships between these two glial cells in the transition zone of the Ranvier node. And we see a much richer environment with the terminal part of the myelin in green. In violet there is the axon, and in yellow is an astrocyte. And we can observe that there is a rich exchange of ion channel transporter expression contributing to axonal homeostasis and on the efficiency of the node of Ranvier. Following an action potential coming from image in the left of this figure, it will come in a fast way because we are under the oligodendrocyte and the myelin sheath around the axon. But we arrive to the juxtaparanode region. The juxtaparanode means that there is near the paranode region, that is the region proximate to the node of Ranvier. So we have three stages: the juxtaparanode, the paranode, and the node of Ranvier. And here we have a beginning of ion exchange through ion channels just under the myelin, and myelin in this case acts as a buffering regions beginning to regulate the ionics exchange. And then we have a very important section, because the regulating factors in the juxtaparanode regions of the myelin go through the paranode, and we get to the node of Ranvier. And to prevent a kind of anomalies in the excitation or non-excitation of a potential hyper-excitotoxicity of potassium channels getting out during the electrochemical conduction from the node of Ranvier, the astrocytes act as buffering structures in this gap junction. They keep potassium that is in excess in the extracellular space during the action potential. Let us remember that during the action potential we have a reverse of concentrations of the two ions, while in resting neurons, sodium channels are much more concentrated in the external part of the neurons or axons. The potassium channels are much more concentrated in the first layer underneath the membrane of the cells, of the neuronal cells. But during the electrochemical transition of the signal, we observe a reverse of concentration, and we have an excess of sodium ions in the internal of the cells along the node of Ranvier, and an excess of potassium outside the node of Ranvier. These excesses must be buffered, must be regulated, and we have this buffering regulation by, for instance, the astrocytes that keeps and gets into the excess potassium ions, and there are also these blue structures that are representing a kind of pumps getting out 3 ions of sodium and getting back 2 ions of potassium within the node of Ranvier. and just as the signal is passed, the node of Ranvier must become again quiescent, stable, with potassium ions much more concentrated in the inner part and sodium ions in the external part, just because it must be ready for the passage of a second or following signal. So we have a very dynamic and very complex exchange of ions, density of ions, buffering structures like the astrocytes are able to do, and buffering also structures in the juxtaparanodal region of the glial oligodendrocytic myelin to maintain the node of Ranvier in perfect efficiency. The major myelinization in its functional aspects is now represented in a very simpler figure. As we have seen before, we have nude axons conducting action potential by pure electrochemical conductions, so the exchange and the reverse of potential during the passage of the electrical signal with the chemical reverse of concentration of potassium and sodium, as just explained before. And we have also the myelinated axons with the different lengths. We are showing here almost the same length myelin sheets around the axons, but we must remember that there are very long tracts of myelin in deeply myelinated axon with thick myelination processes and less myelinated axons. And we have also here the nodes of Ranvier, and we can see the region of electrochemical conduction signed in the node of Ranvier with plus and minus, and the spread of depolarization signed also by the red arrows with the electrotonic conduction. Obviously, the thicker and longer is the myelination around the axon, the faster will be the conduction between, I repeat here, the conduction under the myelin is an electrotonic conduction, and is sustained by electrons, a very fast conduction, while obviously the electrochemical exchanges are much more slower. And we have finished here the oligodendrocyte section, and we must go to the other leading actors in the glial constellation, and we will go through astrocytes and multiple roles, the astrocyte lineages, their embryology, and their developmental features. Astrocytes are the most represented glial cell type in the brain. With oligodendrocytes and microglia, the glial cells are more numerous than neurons in the brain. So let's keep in mind that when we speak of brain per se as a structure, we speak in strong amount of astrocytes, oligodendrocytes and microglia. There are studies, recent studies, that gives the amounts of glial cells in comparison to neurons, and it appears that astrocytes and oligodendrocytes are 1.2 more numerous than neurons. Once upon a time, it was thought some years ago that they represented 10 times the neurons, but now the percentage has dropped drastically. And from the simple role of sustaining cells for neurons, as was envisaged by Rudolf Virchow, just as the second image of this talk, astrocytes have revealed a crucial chemical, biochemical, metabolic, and supporting role for neuronal networks, and more, for the whole central nervous system. I quote here a colloquium that I had with Mark Noble, and it was 1984, so many years ago. Mark Noble was a star of the glial cells studies in the world, and it has been one of the most remarkable neurobiologists at his times. And he remarked to me in a personal communication, he was passionate with astrocytes, that astrocytes can be considered the unsung heroes of the brain. And it is a beautiful remembering for me, because it gave me the right measure of the astrocyte and oligodendrocyte thought. All my career has been built on neuronal recordings. And now let's have the same picture that we examined many pictures ago. It is just to show to you that at the end of the neurogenetic process, we have the stage of the true glial process with the oligodendrocytes and astrocyte generation from precursors, astrocyte precursors in AP and oligodendrocyte precursors in OPC indicated. And generation, they exhibit generation, differentiation, and maturation stages with different time profiles among different brain regions. So the astrocytes do not mature in the same time, but they mature in further stages, in some subsequent stages, in steps. Usually this happens after the main periods of mammalian neurogenesis and neuronal migration. And we can see in the red enclosure the stages of this maturation of glial cells. There are few regions escaping from this law that are the hippocampus and the subventricular region of the cortex, but it is not such important here. Even though all astrocytes share common properties, recent physiological and gene expression profiling studies revealed that astrocytes show more morphofunctional features than previously assumed, with distinct properties in different brain regions at different periods of development. And we can see here an astrocyte that has important roles cooperating in the production and the distribution of antioxidants, and to colloquiate by special channels with little vessels and capillaries, and they have also a blocking action on the toxicity pathways coming from protein aggregates as A-beta or alpha-synuclein that are specific of Alzheimer's disease. The groups of astrocytes have been recognized on the basis of their morphology and location. There are fibrous astrocytes of the cortical white matter. Fibrous astrocytes are associated with myelinated axon tracts such as the corpus callosum of the cortical spinal pathways from the motor neurons, and are in contact, as we have seen before, with the nose of Ranvier. The second group is represented by protoplasmatic astrocytes, and they are specific of the cortical gray matter, and they are highly ramified to penetrate the surrounding tissue, because they must find their path among the neurons that are amassed in the cortex. These astrocytes ensheath neuronal synapses. They are extremely useful for synapse functioning through their fine perisynaptic processes. So while fibrous astrocytes are associated with myelinated axonal tracts, and they are supporting the myelinated axonal tracts, the oligodendrocytes involved tracts of axons, and at the nose of Ranvier and elsewhere, the protoplasmatic astrocytes ensheath neuronal synapses. So it is very easy to understand that their role in the cortex, just because they are very highly ramified and they must find their path among neurons, they must have some other roles than fibrous astrocytes. And in fact, they have a very fine role in perisynaptic space regulation, stemming from secondary and tertiary branches, giving these cells their characteristically intricate bushy structure. Protoplasmatic astrocytes are also in direct contact with blood vessels through a specialized cellular compartment called the hand foot. And we can see here the two types of astrocyte, but we must go a bit in depth in the next figure, where we have fibrous astrocytes that are supporting the myelinated axons and defining and their counterpart of oligodendrocytes, myelin, and sheathing the axons, and the protoplasmatic astrocytes with these different shape and different positioning in the brain, while the fibrous are around the myelinated axons. The protoplasmatic astrocytes take care of the synapses and the vessel, and we will see how. We have again the same figure that is extremely simplified, but gives the idea of the role of these two kinds of astrocytes. But if we consider protoplasmatic astrocytes that are of particular interest, we can see that we must become aware that the original synapse description that in old ages were described as presynaptic and postsynaptic made structures now must be considered more richly as tetradic structure or synaptic tetrads, that is four parts composing the dynamics of a synapse. And in fact, we have into the rounded rectangle, the space of the protoplasmatic astrocyte and of the synaptic tetrad. We have the presynaptic terminal and postsynaptic terminal, that is the dendrite of the receiving neuron, the signal receiving neurons, that is identified by this violet oval. Then we have a very important region that is the glial synaptic hinge, or the blue circle here. And it is composed of presynaptic, postsynaptic terminals, that is the synaptic terminals of the upstream incoming neuron fiber, the postsynaptic or downstream dendritic terminal of the connected neurons, and the astrocyte terminals surrounding the synapse. This is a crucial zone because the synapse can remain healthy and well-functioning because of the rich exchange of substances and transmitters and proteins with the astrocytes that are in some way the nurses of these synapses. And the full description of this ensemble would be even more complex. We will see something in the next figure. And then we have the astrocyte terminal branches getting in touch with the cerebral microvessels in Page's circle by establishing very complex anatomo-functional relationships with the vessels themselves. And this big region with 4 components is crucial. And it is a tetradic synapse, not a dyadic synapse, as in older texts and older descriptions. The so-called triadic synapse of astrocyte and pre- and postsynaptic terminals must be enriched by the relationships that protoplasmatic astrocytic terminals with blood vessel entertain, becoming a tetradic synaptic complex. The structural and metabolic side is a synaptic tetrad. Perisynaptic astrocyte processes are structural and functional components of synapses. We can see here a fibrous astrocyte just enclosing, just imaged, in these fluorescent light blue dye protoplasmic colorant or dye on a protein associated to the membrane of the protoplasmatic astrocyte in the mouse cortex, and observe the impressive ramification, the morphological complexity of an astrocyte in vivo. It is a fluorescent light blue dye on a living astrocyte in culture. And in B, we have an electron micrograph of the synapse, where it has been taken the mouse visual cortex, visual area 1 or V1, the pre-synaptic or axonal synaptic terminal, and the postsynaptic or dendrite terminals are indicated by reddish and greenish colors. And we can see the extremely important element indicated by astros, that is the protoplasmatic astrocyte enclosed, pushed within the neuronal cells, getting its prolongation at the border of the synaptic cleft in the higher part of the figure. There is a prolongation of the astrocytes, almost closing the synaptic space between axon and dendrite. And we have here, and the astrocyte is indicated in azure, in light blue. And we have here in C a schematic design of the presynapse, the postsynapse, and the neuromodulation played by the astrocyte terminals. Astrocytes are functionally crucial for the synaptic homeostasis, as we saw before in the previous slides. In addition, they possess the ability to sense the synaptic activity. In fact, if you look at the presynaptic terminal with the synaptic vesicles containing the transmitters or the modulators in blue getting out from the presynapse, some molecules of these transmitters or modulators, usually taking relation with the postsynaptic receptors on the dendritic or somata postsynaptic terminals, and starting the new passage of the electrochemical signal. These molecules sometimes go out of the synaptic cleft and are able to activate specific receptors on the surface of the glial cells in blue. And these neurotransmitters can give rise to important responses of glial cells, and in the meantime, neurotransmitters granted by glial cells can modulate the presynaptic action towards the postsynaptic terminal. And we have also the calcium intervention, because the astrocyte possess the ability to sense the synaptic activity and response, not only through these receptors of neurotransmitters, but also by calcium transients and by releasing neuroactive molecules that can signal back to the presynaptic terminals. This means that we have a complex circular metabolic loop continuously running in the astrocyte synapse, astrocyte vessel, astrocyte synapse again interrelation. And this is an image that I don't pretend absolutely to explain in details, but just to show to you how the metabolism of neurons and of astrocytes with the bordering blood microvessels and capillaries are able to put in act for a correct function of the neuron-astrocyte colloquium. And this means that a complex circular metabolic loop is continuing in the ASAP interrelation, as we have seen before. And it is extremely important to describe them, but it is useless because it would need at least 1 or 2 hours to describe analytically this. So you must take them as granted, and to understand the extreme complexity of the astrocyte roles that cannot be described with the analyticity of the oligodendrocytes. Through specific physical neural terminal process, synaptic surround, protoplasmic astrocytes control the formation, maturation, activity of synapses by sensing neuronal inputs and intermodulating neighboring synaptic elements through various mechanisms such as uptake or release of neuroactive factors. And I show here the counterpart, the living counterpart of the previous figure, and do not be worried to understand in the particulars of these figures, but imagine that we are looking at the green and the blue and the red parts. The blue parts are the internal of the vessels, of the little vessels, and the green part are the protoplasmatic astrocytes containing these vessels and exchanging proteins, substances, oxygen, and so on to be sent to the neurons, as we have seen in the previous factors. And they regulate the perivascular homeostasis, the blood-brain barrier, BBB integrity, the crosstalk with the peripheral immune system, endothelial transport, the vessel contractility, and the response to neuronal activity. Let us keep in mind that when a neural signal comes to the postsynaptic neurons and the astrocytes sense them, the astrocytes communicate with the vessels, and they provoke the dilation of these vessels and permits to substances to transit from the vessel to the astrocytes, and the astrocytes will have the task to distribute nutrients and feeding substances as well as oxygen to the synapses. And synapses will give back the signals of the received signals to the astrocytes that will communicate it to the vessels that will regain their original diameter. So we have dilation of vessels because of the astrocytic signaling activated by the neuronal activity, but they are not contemporary processes. And in fact, there are also different times characterizing the activation of neurons and that of astrocytes. Sometimes astrocytes are overfilled with some substances and do not signal particularly to vessels that remain with normal diameters, and they can stay, not to say inactive, but little active in the exchange with astrocytes. Sometimes the astrocytes signal very heavily to the vessels that becomes very dilated. And this is the principle of the so-called functional magnetic resonance that indicates the so-called blood level oxygen in the brain areas. But as we have noted before, the dilation of a vessel is not the obligate step by the activation of astrocytes, because sometimes astrocytes are very well fed and can communicate with neurons independently from the vessels. So all the images we get from fMRI, the functional magnetic resonance, have to be kept and have to be considered with much patience and much circumspection, because there is not a direct causal effect of neuronal activity and vessel dilation. The simplified explanation of the image: the astrocytic terminals have a plethora of biochemical heavy elements. And as we have seen, we have metabolic, genetic machineries within the astrocyte amphitheater with messenger RNAs and so on. And this is a repetition in better details of the previous image, but it does not say particularly more than the previous. People interested can go reading through this text and getting to the end. Let's give just a snapshot on microglia and on its roles and interactions with macroglia, astrocytes and oligodendrocytes. And we can see here the complex interrelationships between astrocytes, microglia, blood vessels, and neurons. Astrocytes and microglia provide an homeostatic support for several neuronal functions. Astrocytes recognize neurotransmitters through receptors of glutamate, of GABA, and adrenergic molecules that provoke different modulation in intracellular calcium, as we have seen, inducing responses to neuronal synaptic activity, and most importantly, they interact with microvasculature, allowing the complex metabolic thread, as illustrated in the previous slides. Anomalies of astrocyte calcium-dependent signaling leads to macroscopic pathological events, both in metabolism and behavior. Microglia is strongly involved in these metabolisms and regulates neuronal activity in synapse formation after contact with dendrites. They too, in the formation of spine and head filopodia. What does this mean? That microglia is essential for the construction of well-functioning dendritic spines. As we know, dendrites are constellated by little bumps that represent the point of synapse with the incoming neuronal signals, and these bumps are called the dendritic spines, and microglia contributes heavily with feeding factors to the well-maintenance of these bumps or glia or dendritic spines, and they intervene, as we said, in the formation of spine, head, filopodia, and in many aspects of head filopodia. If we can observe, as they did very strictly in two-photon microscopy, the nascence and the death of this dendritic spine, we can see that they are very fast processes, continuously changing the receiving properties of the neurons and continuously regulating the receiving properties of the neuron itself. And they go up and down, and they are modulating the so-called neuronal plasticity. That happens just with the cooperation of astrocytes, microglia, and blood vessels. And we can see here the different colloquium between astrocytes and microglia, the different shapes and roles played by this duo dynamics in health and disease, the function of the astrocyte microglia module from homeostasis to pathology. Under physiologic conditions, astrocyte in ochre, in light maroon, and the microglia in blue support neuronal functions, where in pathologies they lose some of their supportive function in favor of optimizing the simple survival of these cells. This is accompanied by a change in morphology. If we confront the highly branched and the hypertrophic, structurally rigid phagocytic astrocyte, we can understand the sufferance of the astrocytic cell, the third cell indicated here in ochre, that changes radically its interrelationship with the microglia, that becomes, instead of ramified, becomes with small prolongements, and there is a change in morphology, secreton, that is the substance secreted by these cells, and the increase in reactive markers or phagocytic reactivity. The microglia favors the phagocytic activity of macrophages that eat and destroy the neurons. And this explains why the damages of the astrocytes and microglia can induce devastating damages in the neuronal well-being and correct. The CNS immunity that is maintained by the microglial cells is better indicated here in the 4 rectangles, where we see how the brain parenchyma is surveilled by the perivascular macrophages and microglia. And we have different factors that show the engulfment and the regulating roles of microglia. This involved the engulfment and destruction of neural precursor cells, the MPCs, the A in the A rectangle, with the engulfment of these neural precursor cells that are included within the macroglia and then distributed to the macrophages. And then there are other degenerative processes that are conducting to the engulfment and the destroying of the degenerating weak synapses. This could be also a positive factor, because during the day we accumulate an enormous number of active synapses that are not essential for the well-being of the neurons, but in some way they outnumber the quote of correct synapses populating the brain. We can have too many synapses and they must be destroyed. And this destruction is not always a negative effect, but it can serve to the sculpting, the correct sculpting of the synapse, favoring the strong synapses and destroying the weak and non-serving synapses. This happens, for instance, in many stages of sleep, where weak synapses are destroyed and eliminated by the cooperation of microglia and macrophages. And we have also other actions, like pathologic response as an acute, sorry for the double C, response to tissue damage as in B, with increased ATP formation that toxicate the synapse, and in D we have the homeostatic surveillance of healthy, strong synapses, and that gives rise to the elimination of increased potassium in the synapse space. And we have other acronyms with NPC, MERTK, complement component 1Q, CD11B, P2RI12, and THIC1 factors. And you can read very simply them. They are not particularly important to make a list and to explain name by name. As we have learned from previous figures, the enormous number of regulating factors is continuously made by proteins, glycoproteins, lipoproteins, and they, in this case, they enlarge the number of these regulating factors regarding also microglia. I put here 2 images for retinal glial Müller cells, not because I want to show you something new on Müller cells, but because the previous long list of slides can serve to you as group of ophthalmologists interested in this kind of glial retinal cells and suggesting to you different paths for making research on this interesting section of the glial retinal cells. I show here a for you useless image showing in light blue the so-called Müller cells that goes from the surface, inner surface of the retina to the apical surface of the retina. And in the inner surface we have the nerve fibers getting together throughout the cribrate membrane of the eye, reuniting them in the optic nerve, and on the surface of the retina, that is the deep, in truth, the deep layer of the retina, we have the cone and the rods and cones represented in the superficial stages with amacrine cells, and then bipolar cells, rods, cones, ganglion cells, horizontal cells, microglia, and Müller cells. The different layers of the retina are defined in their acronyms in the figure, in the explanation, in the written part of the figure. But I don't think that any one of you needs of these explanations. And the last, and very last image shows that Müller glial cells can resume proliferation in the rodent adult injured retina, but contribute weakly to tissue repair when compared to zebrafish retina, as we have seen in the previous slides. Like the macroglial component of the brain, also the retinal major glial component, such as the Müller glial cells, undergo the effects of markers and regulatory factors, such as the ACSL1, archi-acute amyloid 1, that we have already met before. But there are deep differences, and many of the factors taking care of the well-being of Müller cells are not known. And this is an interesting kind of scientific program that can occupy your future researches on Müller cells and on other glial retinal cells. But this amyloid 1, SCL1 factor, has an important role in that it can reprogram the Müller cells in neurons that are called induced neurons, that show features analog to different types of visual retinic cells, such as photoreceptor, bipolar, and amacrine cells. Thus, reprogrammability may be listed in the features of these Müller cells. For instance, TF-neurogranin 2, another factor, regulating factor, present in the developing retina, is also sufficient to lineage reprogramming postnatal mouse Müller glial cells into the new neurons. And it is extremely an important steps. The glial to neuron anatomal functional shift can be enhanced after the expression of the ACSL1 or NeuroG by EGF protein that stimulates cell growth and differentiation by binding to its receptor, the EGFR, and the FGF, the fibroblast growth factor, that has shown potential effects on repair and regeneration of retinal tissue besides the other tissues. This is partially explained by an increase in the frequency of Müller glial cells expressing sex-determining region Y, SRY box 2 or SOX2. We saw the SOX1 in the previous slides when we spoke about oligodendrocytes in the previous slides. Remember, comparative factors are present in the development of macroglia in the brain, but not all the factors are the same, and big surprises are waited in the future of the studies of these retinal glial cells. Thank you for your kind attention and for following me until this late hour.